Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Probing and Disrupting Oncogenic Gene Control via Targeted Protein Degradation. This webinar is sponsored by Lexogen. Our speaker today is Dr. Georg Winter, Principal Investigator at CeMM, the Research Center for Molecular Medicine of the Austrian Academy of Sciences in Vienna. You may type in a question at any time during the webinar, and you can do this through the Q&A panel, which normally appears on the right side of the webinar presentation. We will ask Dr. Winter your questions after his talk has concluded. Also, if you look at the bottom tray of your window, you will see a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Winter. Please go ahead.
Thank you, Julia. It's my great pleasure to be here today and give you updates about our latest research that all center around trying to probe and understand gene control in cancer using a new type of pharmacology called targeted protein degradation. Before we get started, a brief conflict of interest statement. I'm founder and shareholder of two recent spin-offs, Proxygen and Solgate, and I'm also coordinating a sponsored research collaboration between our research center, CeMM, and Pfizer. In general, the research focus of my lab is really centered around a couple of different topics, being mostly chemical biology, transcription, the science of gene control, and cancer. And so we're really excited when all these three disciplines meet each other. And so particularly we're interested in using chemical biology to probe cancer cells to derive novel therapeutic insights, but also to use chemical biology as a means of acute perturbation to perturb transcription at a high kinetic resolution to allow us to derive first-order insights into mechanistic biology. And I'll try to share examples of both of these two strategies that we're employing. One particular challenge when conducting chemical biology at the interface of gene control is the fact that only 20% of the proteome can be explored with small organic molecules. And the reason for that is really how we traditionally have defined druggability. So what you see here on your right is a depiction of the EGFR kinase domain as a prototypical druggable target. So we think about a druggable target as having a hydrophobic pocket that encodes some sort of biochemical activity that we can fashion small organic molecules that bind said pocket, and it's kind of lock down that encoded biochemical activity. And as a consequence, we call these type of proteins druggable. The downside is that the vast majority of all proteins encoded in the human genome lack such an accessible, catalytically active and hydrophobic pocket. Prime example would be, for instance, transcription factors, as seen here with a micmax heterodimer sitting down on DNA. I want to conclude on that slide by saying that this very inhibitor-centric mindset and traditional way of how we think about druggability has really limited that interface between the therapeutic science and the science of gene control. So what are alternative means to the goal of pharmacologic control of gene control? And I think one of the most intriguing examples of that is given to us by nature. So what you see here is in that cartoon is an E3 ligase, a plant E3 ligase, that incorporates the tier 1 ligase substrate receptor. And what happens is that when a small plant hormone, a phytohormone called auxin, binds this tier 1 substrate receptor, we induce the proximity between that ligase and a family of the transcription repressor proteins called IA7 proteins. And the result of that induced proximity to the degradation machinery is the ubiquitination of IA7 and its ensuing degradation by the proteasome. So induction or inducing proximity to the degradation machinery could be a viable option to the therapeutic science at the interface of chemical biology and gene control. In its first iteration, or first chapter, we have been working together with other pioneers in the field like Craig Cruz or Alessi Zuli on heterobifunctional molecules that basically phenocopy the principle of auxin. namely the induced proximity between a protein of interest and an E3 ligase. And they do so by having two warheads, two binding moieties, two arms. With one arm, they bind to the E3 ligase, to the substrate receptor. With the other one, they bind to the protein of interest that we want to get rid of. And so the result of this dual binding event is an induced proximity, which again results in target ubiquitination and degradation by the proteasome. These PROTACs are relatively straightforward to design when there is a binding ligand available to a protein of interest. And importantly, because they have catalytic turnover, they can be incredibly potent. Catalytic turnover here means that we can induce the degradation of more equivalents of target proteins than we have of small molecule. Importantly, we could show that this therapeutic concept holds up in vivo. And over the last 5 years, there has been tremendous progress in that field, leading to the first clinical trials that are currently pursued, particularly for androgen receptor and also, I believe, estrogen receptor targeting degraders. So with that brief background, I want to tell you a bit, I want to give you an outlook of the two projects that I want to discuss with you today. The first one is where we're trying to use transcriptomics and functional genomics to expand the toolbox of degraders, find novel type of degraders that induce the degradation of members of the gene control or gene control proteins. And the second chapter I want to share with you is where we are using targeted protein degradation as a very focused means to understand a protein complex called the Mediator complex that is also involved in transcription control. So I want to start with the first one. In order to set the stage, I also want to tell you that PROTACs, despite we all like them a lot, also have certain limitations. And one of the limitations really stems fundamentally from its concept of a dual binding mode. Because that means, in other words, what that basically means is that we can degrade what we can also bind, or we can degrade what we can ligand. So I think it's fair to say that in the literature there's no evidence of a case where PROTAC could induce the degradation of an unligandable protein. And so particularly if we again think about transcription factors, this poses a certain challenge. There's other challenges like the high molecular weight and also only a limited number of E3 ligases are amenable. But I think for our purposes, really this inability of degrading unligandable proteins is probably the biggest hurdle. Luckily, there's a way around that as well. And we believe that this is presented by the so-called molecular glue degraders. So what is a molecular glue? Auxin, what I've shown you in the beginning, would be a molecular glue. Other molecular glues are these analogs of the thalidomide molecule. So here represented by the anti-myeloma drug Revlimid. So molecular glues also induce the proximity of a protein of interest and an E3 ligase substrate receptor, but they do so by inducing a very cooperative or highly cooperative protein-protein interaction. And that allows molecular glues to induce the degradation of target proteins that would otherwise be considered unligandable. They can degrade proteins without actually binding to them. They're clinically already validated, as seen by the highly successful drug Revlimid. But the big problem in the field has been that the discovery of those molecular glue degraders or molecular glues has been highly serendipitous. And this is something that we wanted to change in my group. So the first step towards changing that was to understand all cellular effectors that need to be active and need to be in place in order for target-induced degradation to work. In order to map these cellular effectors, we conducted 5 genome-wide CRISPR screens with different PROTACs and also molecular glue degraders. So what we did, we took near haploid KVM7 cells expressing Cas9, transduced them with a genome-wide library, applied selective pressure by titrating in drug, in this case PROTACs or glues, selected for surviving clones, and identified the mutated gene by sequencing the guide RNAs. And then in order to get an overview of all of these effector networks, we mapped all effectors that we identified on the protein-protein interaction networks, and that's what you see here on your right. And basically, the quick take-home message here is just that we find effectors that are highly drug-specific, but we also find a very dense interaction network of effectors that appear to be relevant for the vast majority of all PROTACs and glue degraders. There was in particular one gene called UBE2M that appears to convey universal resistance to all of these PROTACs and glues that we tested. So what is UBE2M doing? UBE2M is a gene that is involved in the nedylation of chitin ring ligases. So here you see again a cartoon on your left where chitin ring ligase is active when it is nedylated. Excuse me. So when we investigate the activity or the nedylation status of chitin ring ligases using Western blot, we basically always see two bands. So this is you would see that in that Western blot labeled in the wild type column, where the higher molecular weight band would be a nedylated chitin backbone representing an active ligase complex, and the lower band would be the hypo or unnedylated chitin backbone representing an inactive ligase complex. So this post-translational modification of nedylation is something that we can block with a small molecule inhibitor that is relatively toxic, here indicated as by its name, melinum-4924. And when you see when we treat those cells, we see a global denedylation of all chitin backbones. Interestingly, we see a very similar feature in those cells that we have identified to carry a hypomorphic allele of this UBE2M gene. So again, you see that the vast majority of all chitin backbones is present only in its unnedylated form, and we can also reverse that by reconstituting this mutant clone with cDNA encoding for UBE2M. The important difference to the pharmacologic inhibition of that pathway, however, is that those cells grow just fine. They grow exactly at the same kinetic as its wild-type counterpart. If we do an analysis, transcriptome analysis of these particular KBM7 cells, when we ask the question, how many of these different assemblies of chitin backbones or chitin ring ligases are expressed, we can hypothesize that this particular UBE2M mutant allele affects the activity of up to 220 different chitin ring ligases. So what can we do with these hyponedylated cells? To circle back, what we know of those cells is that when we mutate UBE2M, they confer resistance against all known degraders. So what you see here is a dose response curve of KBM7 wild type and UBE2M mutant cells treated with a BAT4 PROTAC called DBAT6. And you see that the mutant cells are a factor of more than 100 more resistant. So we thought we can maybe also turn that question around. And instead, using this cell line pair, testing thousands of molecules that have no associated or annotated function, and specifically search for molecules that would kill wild-type cells, but that lose activity in the absence of active UBE2M. And that the result of that chemical screen you see in this scatter plot, where you see that we identified a small set of molecules that fulfill just those criteria. And none of them had a known function, and all of them validated in dose-ranging experiments as seen on the right. So in the following, I want to show you a couple of those examples that we worked up. So at this point, so what you see here is a molecule that we pulled out of that screen that we very creatively called DSM1. And what you see in that cartoon is basically a depiction of the status quo at that moment. So we just knew that a chitin ring ligase is involved in the activity of that molecule. We didn't know what ligase, and we didn't know how the ligase is reprogrammed in order to induce degradation of what kind of protein. The situation around DSM1 was a bit particular because based on its chemical structure, we had a hint. And the hint was delivered by an excellent paper by Ting Han and al., where they have identified that aryl sulfonamides around the compound shown here, called indisulam, can act as molecular glue degraders. Pretty similar to the so-called IMiDs, or these thalidomide analogs. How these aryl sulfonamides work is that they bind an E3 ligase called DK15 and rewire it in order to degrade a splicing factor called RBM39. So here we had kind of a working hypothesis what DSM-1 would be doing. When we used here, what we did here is we used a focused CRISPR library that allows us to mutate all culinary ligases and all known effectors. And again, we can ask the question, which genes convey resistance to DSM-1 treatment? As predicted, the most strongly enriched gene that we find is that DK15 E3 ligase. Again, we can knock out DK15 as well, and we see that the activity of DSM-1 is rescued. You see that in the dose response curve in the middle. Our molecule, similarly to indisulam, induces the degradation of RBM39, as seen in the Western blot, and that degradation is DK15 dependent. So again, if we mutate DK15, we don't see destabilization of RBM39. And we could also show that the degradation of RBM39 is highly selective, as shown here by expression proteomics. So at this point, we were excited because we could show that with this strategy, we can identify novel chemical matter that phenocopies the molecular glue degrader mechanism, or a molecular glue degrader mechanism that has been established in the field. But what about all the other scaffolds that we identified? So the story I'm going to show you centers around these three molecules, DSM2, DSM3, and DSM4. At this stage, again, we didn't know what is the ligase involved and what is the target that is degraded. And here it was a bit trickier, so we did a couple of more experiments. The first experiments that we did was an expression proteomics experiment. So again, we treated leukemia cells with these three different scaffolds for 5 hours and just asked what is the protein, the overall change in protein abundance. And interestingly, despite the kind of different chemical structure of these molecules, all of them induced the degradation of cyclin K and a minor but still measurable destabilization of the cyclins CDK12 and CDK13, which are the two cyclins known to associate with cyclin K. We could validate that cyclin K degradation also by immunoblot for all of the three molecules, as you see below the proteomics data. Next, we used transcriptomic profiling in order to understand basically whether degradation of cyclin K by our molecules phenocopies catalytic CDK12 inhibition. So we made use of a highly selective CDK12-13 covalent inhibitor called THC531 from Nathaniel Gray's lab, and we treated leukemia cells with that inhibitor or with our 4 different glue degraders, where DSM2, 3, and 4 we knew would degrade the associated cyclin. DSM1 we knew was the RBM39 degrader. And then we used gene set enrichment to ask the question, how similar is CDK12 inhibition to the perturbation inflicted by DSM2, 3, and 4? And the answer was that it was highly similar and highly correlated. Importantly, what we did here is that we also, because we knew that both CDK12 and CDK13 are involved in a process called transcription elongation. So we anticipated that either blocking its catalytic activity or degrading its associated cyclin could lead to a global disruption of mRNA abundance. So it was important for us here to use synthetic mRNA spikins that allowed us to do all of these measurements in a cell count normalized manner. And what you see here in these volcano plots on the left is basically that both catalytic CDK12 inhibition and cyclin K degradation lead to global disruption of mRNA abundance. We could presumably have made very similar observations if we would have used nascent RNA sequencing, such as SLAM-seq. But for us here, as an initial transcriptional profiling, spiking normalized RNA sequencing was very insightful technology. Next, we did a couple of rescue experiments. So what you see here again is cells treated with DSM2 with the left of that Western blot, and we just co-treat with DMSO. We just see that cyclin K is degraded. Very expectedly, we could rescue that degradation by blocking the proteasome with carfilzomib, by again perturbing the methylation cascade, by treating with MLN4924, by blocking E1 with TAC243. But most interestingly, we could also rescue cyclin K degradation when we co-treated or pre-treated with this covalent CDK12-13 inhibitor THC531. So that insinuated that we need an available active site on CDK12-13 to induce cyclin K degradation. And we could validate that our molecules in fact also bind to CDK12 and 13 by teaming up with Matthias Geyer's lab, who ran a recombinant 12-13 kinase assay, which showed that while not very potently, nevertheless our molecule bind to CDK12 and CDK13 and also mildly abrogate its activity. So at this point, we were relatively convinced that our molecules would induce the degradation of cyclin K. The question was how. And so in order to understand that, we again used our Cullen ring ligase-focused CRISPR guide RNA library. And again asked the question, knockout of which genes conveys resistance to our cyclin K degraders. And what is very interesting here is that we find super strong enrichment of only a handful of genes. And if we look closer, this handful of genes basically make up one entire but incomplete Cullen ring ligase complex. So we find a Cullen backbone, Cal4B. We find an adapter protein, DDB1. We find, again, UBE2M, one of the genes involved in the methylation of Cal4B. And we find UBE2G1, which is an E2 ligase that associates with the Cal4B backbone via RBX1. The big surprise was, however, that we did not find a dedicated substrate receptor. We did not find the cereblon of this particular ring ligase complex. So the first assumption that we had is maybe there is an orphan substrate receptor that was not part of our focused library. So we repeated the entire screen, this time using a genome-wide screen. But to our disappointment, or still excitement, we found exactly the same 4 genes. So again, no dedicated substrate receptor was in sight. So we concluded from that that the induced degradation of cyclin K might be substrate receptor independent. To gain further confidence in that, what we did is that we took wild-type cells, again KBM7 cells, and exposed them to high levels of our cyclin K degraders. We then selected for viable cells, and we performed a targeted sequencing of 50 genes that were informed by our genome-wide CRISPR screen. Among them were also Cal4B, DDB1, and other genes that we identified in the CRISPR screens that I've just shown you before. And very interestingly, what we found is that the vast majority of spontaneously arising mutations that convey resistance to our cyclin K degraders cluster in DDB1. In particular, they cluster in the BPC pocket. And when we map all of the mutations that we identify on a known crystal structure, it was very interesting to see that they all line a pocket that is hijacked by also a viral peptide. So for you to know, viral proteins also sometimes hijack the cellular degradation machinery of its host cell in order to induce the degradation of unwanted host proteins. And so the mutational pattern that we have identified here gave us yet another layer of evidence that maybe our small molecules phenocopy that mechanism. So instead of hijacking and reprogramming a substrate receptor, maybe they reprogram just the adapter protein DDB1 directly. In order to validate that hypothesis, we teamed up with Nico Thomas' lab, who have developed a very nice TF-RET assay that allowed us to really take these mutational patterns and reconstitute the entire system in a fully recombinant manner. So the TF-RET assay consists of recombinant DDB1 and recombinant CDK12 cyclin K. And so what we can now do is we can titrate in our compounds and ask the question whether compound addition induces proximity of these 2 recombinant proteins. And the answer was yes. We did see that also in the absence of drugs. So just when we treat these proteins with vehicle control, there's a certain baseline affinity. However, upon treatment with our DSM2 molecule, we vastly induce the proximity of those 2. So that brought us to a model where we hypothesized that our molecules might act via a unique and very non-obvious mechanism of action where they bind to the CDK12-13 active site. And that binding event induces the proximity between CDK12-13 and its associated cyclin K to the DDB1 that is part of this impartial Cullen ring ligase complex. And so in a way, CDK12 and 13 almost mimics the activity of a substrate receptor, presenting cyclin K to the associated E2 ligase UBE2G1, which induces its ubiquitination and degradation via the proteasome. Importantly, right around the time where our study was published, there's two other papers that I want to mention here that describe a very similar gluing of CDK12 cyclin K to DDB1. So this really appears to be an emerging mechanism that we tapped into. In particular, the study of Nico Thomas and Ben Eber's lab also solved the crystal structure of non-CDK12-13 inhibitor that very similarly induced the ablation of cyclin K in cells. So this inhibitor is called CR8, and interestingly, they could also identify the structure-activity relationship, pointing to that pyridal moiety that is sufficient to induce the proximity between DDB1 and CDK12. And there was also a second paper published in eLife by Tin Han's lab that also reported on new chemical matter, similar like our paper, that induce cyclin K degradation. Okay, so far I've told you our efforts to really expanding the glue toolbox or the toolbox to approach novel therapeutic strategies to induce the degradation of translationally relevant gene control proteins. However, we think that protein degradation can also help us to really understand transcription mechanisms or any kind of a mechanism for that matter. And the reason why we think so is that we believe that protein degradation matches up very well with the known perturbation strategies that we mostly apply in the lab. And so these traditional perturbation strategies are either genetic approaches or traditional pharmacological inhibitors. So advantages of pharmacological inhibitors is that we have a certain dose control, they are typically nicely reversible, and they have a high kinetic resolution. So we can perturb and then measure relatively fast. The big disadvantage that they have is that A, they're very limited in terms of the scope. We can't inhibit every protein. That's kind of where we started. And then the second big disadvantage is that if we can inhibit the protein, we only inhibit one discrete function. We only inhibit the kinase function of a kinase, but we don't block its scaffolding function, or we don't block maybe its SH2 domain. And the opposite, if we use genetic approaches, we typically have perturbation either on the mRNA or on the DNA level that in the end result in a global loss of protein abundance. And so with genetic approaches, we can really take out the entire protein. The problem is that typically we don't do that in a dose-controlled manner. It's typically not reversible, and it's typically too slow to really infer direct mechanistic consequences. And so we believe that with protein degradation, there's a very nice niche that allows us to combine the better of both worlds. The problem is that also not for every protein we have a directly acting degrader, and this is why we developed during my postdoc this so-called degradation tag or DTAG strategy as a general solution to protein degradation. It's published. I just want to quickly compare it here. So as opposed to a directly acting PROTAC that you see on your left, where we need a new small molecule, a new PROTAC for every protein of interest that we want to degrade. The DTAG molecule always uses the same small molecule to induce that perturbation. However, you need to have the protein of interest fused to this so-called DTAG or degradation tag, which is nothing else than mutant FKBP12. And so because when you have your mutant protein fused to the DTAG, as here shown for another transcriptional kinase, CDK9, we then can use the DTAG ligand, so this standardized heterobifunctional molecule, to induce its very acute and near complete degradation. In the case that I'm showing here, we have knocked in the DTAG and the N-terminus of CDK9, and you see that in the Western blot, the degradation that we can enforce is both very abrupt and also very pronounced. And so in the lab, we're now interested to apply the degradation tag or DTAG technology to really understand transcriptional control at the high kinetic resolution. Or in other words, we're interested in how the information that is stored in the binding of the almost 1,500 transcription factors all over the genome and how that information is related to the one enzyme that synthesizes all protein-coding genes, RNA Pol II. And one of the proteins that we are particularly interested in is the so-called mediator complex, because it's really a central relay factor in how that information from the bound TFs to RNA Pol II is conveyed. So what is mediator complex? It's a highly conserved molecular machine that in human consists of 30 subunits. And there has been a lot of structural data out there. But there's still a couple of key questions in the field. And the limitation to studying human mediator has always been the fact that it's a highly essential molecular machine. So most of these subunits, of these 30 subunits will be essential. So we cannot generate a knockout cell line. A mediator, maybe importantly to know, is also organized in 4 different modules. But there's also something that is referred to as the core mediator complex. So the key question that we wanted to address using the degradation technology is trying to answer whether individual mediator subunits regulate different target genes. We also wanted to understand whether all genes universally depend on a human mediator. And we also wanted to understand which transcriptional checkpoints are regulated by mediator. So maybe in order to quickly introduce the transcriptional checkpoints, I can go back to this slide. So upon initiation of RNA Pol II, there is one regulated step that is referred to as pausing. So this is when polymerase typically transcribes around 120 nucleotides and then is in this pause state where it waits until it gets a signal through, for instance, by phosphorylation of CDK9, so that it starts a processive elongation. And then that elongation step is finally halted by a regulated termination site or termination signal. So how mediator influences these different transcription checkpoints was also not studied in full. So in order to get a first hint towards these questions, what we did is that we tagged a couple of different mediator subunits endogenously, again using these near haploid KBM7 cells using the DTAG. And so you just see this schematically here in this cartoon. And then again, this allowed us acute and very precise pharmacologic control over mediator subunit abundance. So what you see in that Western blot is just that all mediator subunit that we tagged is basically plotted in based on or as a function of its molecular weight. And you see that always we either treat cells with vehicle DMSO or with a DTAG, and you see that we can, for all of these cases, very nicely induce the acute degradation of these mediator subunits. We could also reconstitute findings from the literature, namely that when we use these cells for microscopy studies, that mediator forms these very dense foci in the nucleus, or nuclear foci. And when we co-stain for MED14 and MED1, we see that these foci overlap. So this would be in the DMSO-treated condition, the upper panel. However, when we then degrade one of these factors, in this case the MED14 backbone of the mediator complex, we very interestingly not only see that we disintegrate the MED14 foci, but we also disintegrate the MED1 foci, so which is a protein that we did not take here, and we also don't degrade it. It's just dispersed overall over the nucleus. And so you see in this cartoon here how we basically imagine that to happen. In order to understand whether these different mediator subunits control different target genes, we employed a high-throughput 3' mRNA sequencing technology called QuantSeq, which is just a very cost-effective and very fast method to basically screen different conditions. And so what we did here is that we collected around 100 of these shallow transcriptomes, which allowed us to test in triplicates the impact of mediator subunit degradation on the transcriptome of the cells, and allowed us to compare those to the impact of degrading known elongation factors, and known factors that we knew that upon degradation we see a global decrease in mRNA production. In this case, we did it by either degrading BRD4 and the bed proteins via a directly acting PROTAC called DBED6, or degradation of the CDK9, the active component of the PTFB complex, either again by a directly acting degrader or via the DTAG. And so what you see really at first sight is if we deconvolute these transcriptomes via principal component analysis, that the mediator degradation, despite its presumed role as a general co-activator of transcription, looks quite different from these pan-cytotoxic or pan-transcriptional bombs like BRD4 or CDK9 degradation. In fact, it appears, if we look here just at average mRNA log 2-fold changes, that the overall transcription changes that are caused by mediator degradation, any of these subunits, is relatively mild compared to degradation of these cofactors CDK9 or BRD4. It also appears as if there is not a target gene-specific transcriptional control, but that it's really all of those components just control the same core set of targets. It is really just depending on which mediator subunit we degrade, just influences the number of genes we are effectively impairing. And not surprisingly, it turned out that degradation of MET14 had by far the vastest or the strongest transcriptional impact. And I say this is not surprising because based on structural studies, we know that the MET14 is kind of a structural backbone of the entire mediator complex. However, what we also have realized from that study, while it gave us a lot of very insightful answers, we also realized that the kinetic resolution, so here we treat for 6 hours and then perform these sequencing studies, the kinetic resolution was not sufficient because when we had a closer look at this principal component 2, it was in fact a MYC transcriptional fingerprint. And when we went back and did immunoplotting for MYC, we could observe that at that 6-hour time point, we have already destabilized MYC levels transcriptionally. So that meant for us that we need to look a bit faster. And in order to look faster, we teamed up with Patrick Kramer's lab in Göttingen, and they performed, or my student went there and performed a transient transcriptome sequencing experiment. So that is basically reporting on nascent mRNA output. So it basically shows us where RNA polymerases produces new RNAs in a nascent manner. And what was very interesting to see is that the vast majority of all genes, as displayed here or as exemplified here by the RAB3 GAP1 gene, don't show a dramatic disruption of mRNA synthesis throughout their gene body. So this would be the rightmost gene track. However, there's also genes such as MYC or MIP here, where upon 1 hour of mediator degradation, we literally see not a single new mRNA molecule produced. So it seemed to be a very asymmetric transcriptional response. And that held up when we stopped staring at individual genes, but just looked at the entire transcriptome, which you see here in that scatter plot. So you see that the median log 2-fold change of all measured transcriptional units is very minor. So we only see a log 2-fold change, a median log 2-fold change of minus 0.29. However, there's a subset of genes that are highly affected. Among those are MYC and MIP, but among those are also other transcription factor genes that form a very densely interconnected autoregulatory network, where each of those transcription factors controls its own transcription, but also the transcription of all other members. So how are these cell identity genes regulated? In order to understand that, we again went back to the study of this nuclear condensate or nuclear foci, for that matter. And what we did here is that we took a clone where we had the MET14 subunit tagged with our degradation tag, and also endogenously tagged Pol IIa, so the large subunit of RNA Pol II. And again, we could show that basically reproducing literature, that there's a strong co-localization in these nuclear condensates of mediator, here measured by MET14, and Pol II. And again, we could show that when we degrade MET14, we also dissolve these Pol II condensates. And so based on that experiment, we thought that maybe Mediator really establishes this or orchestrates these reservoirs of RNA Pol II to basically provide a constant supply for this for the transcription of these cell identity coding genes. So the next step that we wanted to study is really to try to understand the dynamics of these cell identity genes and compare those dynamics to your average Joe gene that is not really affected. And in order to do so, we performed a second nascent RNA sequencing approach called PROTAC, which is a nuclear run-on assay that does not measure RNA throughput as TT-seq, but measures RNA Pol II position information at the near nucleotide resolution. So again, teaming up with Patrick Kramer's lab, and particularly here Björn Schwalb, we could integrate both of those technologies in a manner that allows us for every transcriptional unit to determine two important parameters: the pause duration, how long does RNA polymerase halt at the shortly downstream of the transcriptional start site, and the productive initiation rate, which here measures how many polymerase enzymes can basically make it out of the pause site. And so along that continuum of this pause duration to productive initiation function, we can basically see that the more to the right and the more to the lower end of that graph, we would have very high mRNA output. So if we now map in an unperturbed state all of the transcription units, all genes in this KBM7 set on that graph, what we see is that these autoregulatory transcription factors, those that are highly dependent on Mediator, appear to be very efficiently optimized for high transcriptional output, which means that they have a very short pause duration and they have a very high productive initiation rate. In contrast to that, our regular genes that are based on this TT-seq experiment not affected have a longer pause duration and a shorter productive initiation rate. If we then again degrade MET14 for 1 hour, we observed something that was quite interesting, because we observed that for those highly Mediator-dependent autoregulated transcription factors, we see a drastic decrease in productive initiation rate, which basically means that transcription is shut down at those genes. However, our average Joe gene that was not affected by TT-seq here also was barely affected based on this productive initiation rate. But the reason for that was that at all of those genes, we saw that pause duration was significantly decreased. So that basically means that it appears as if at those genes, there's a buffering mechanism where we have a reduced pausing that basically alleviates the transcriptional perturbation that is inflicted via Mediator degradation. In order to understand what factors are involved in this buffering mechanism, we purified chromatin from MET14 detect cells, again, either plus or minus detect treatment, so either in the absence or upon either when MET14 was active or when MET14 was degraded. We purified chromatin and subjected it to unbiased mass spectrometry analysis, which led to the identification of the fact that all Mediator subunits were very strongly depleted from chromatin, which is not surprising. That goes along with all of our data up to that point. But what was very interesting to see is that the single strongest enriched factor on chromatin is that PTFB complex, the positive transcription elongation factor complex, consisting of cyclin T1 and CDK9. Interestingly, we did not only find that we have more of that complex on chromatin, but also that downstream target phosphorylation, for instance, on serine2 of the CTD of RNA Pol II, also on SPT5 was elevated. So that appeared as if, and again, we only do 1 hour of treatment here, it appears as if there is this very quick buffering mechanism that the cell has basically in the back of their pocket in order to ensure that there's still transcription output at those genes. And one final experiment that we did here in order to functionally address whether CDK9 is really involved in this buffering mechanism, we came up with that experiment where we degraded MET14 by the DTAC ligand, so mediator integrity went down, which means that CDK9 activity went up. In order to understand whether the increase in CDK9 activity had to do with a buffering effect, we blocked CDK9 activity via this highly selective small molecule called NVP2, and that basically allowed us to functionally close that loop, showing that CDK9 is involved in this buffering mechanism. And I'm going to cut it short because I'm at the end of my time. So the take-home message of this second story on a bird's eye perspective is that via this degradation tag or the endogenous tagging of genes allows us acute pharmacological control of their abundance. And when we combine this acute perturbation with fast-acting readouts, such as nascent transcriptional readouts, we can really capture immediate consequences of proteins rather than describing secondary equilibria that have formed after an acute perturbation. And so we think that this is really a technology or a strategy or an experimental setup that is less prone to secondary effects or artifacts. I want to thank quickly people in my lab. It's not the most recent picture, but I want to mention particularly two people. Christina is a postdoc who was involved and performed most, if not all, of the cyclin K degrader studies that I've shown you. And then Martin Jäger is a PhD student who performed most of the mediator work. You see the funding sources, and I also want to acknowledge all the collaborators that have helped us along the way. There's always space for new people, so we still have postdoc positions available in the lab if you're interested in the science of targeted protein degradation and gene control. And with this, I thank you for listening, and I'm happy to take questions.
Yeah, thank you so much, Georg, for this very nice presentation of your work. As a reminder, if you have a question, please type it into the Q&A box. And we would also like to ask our participants to take a brief moment after the webinar has ended to take our exit survey that will be emailed to you and give us your feedback. So let's see what we have for questions here. All right. A couple of pretty general questions. Okay, let's start with this one. Is there a strategy to circumvent the need for an available lysine residue to allow ubiquitination of the target protein?
Yeah, that's a good question. So whenever a protein is not degradable by either a PROTAC or a molecular glue, we always had the feeling that lack of an available lysine on the surface of the target protein could be among the reasons. I don't know how often that really is the case, because we know from the Calin ring ligases that they're quite sloppy enzymes. So they have a relatively wide zone where ubiquitination can happen. So I'm not aware of too many studies where a single ubiquitin site or a single lysine residue was really sufficient or necessary for induced degradation. I think there is one. I can't think about it in the moment, but it's not. I don't think this is a general rule. So I don't think that this is the prime issue that we need to solve. I think most of the time it's really formation of a stable and productive ternary complex.
Mm-hmm. Okay.
I hope this answers the question.
Yeah, thank you. Other question we got is it possible to use a single-chain monoclonal antibody as a ligand?
So I assume this is for a ligand that binds to the protein of interest.
I think so. Mm-hmm.
Yeah, I would say in principle, yes. I think the question, as always, is how do you make it cell permeable so that, in the end, you probably want it to be intracellularly available.
Right.
There has been a lot of very interesting work with nanobodies and other engineered systems. So I think in principle it's perfectly feasible. To us, whenever possible, we try to go for a small molecule solution.
Yeah. Question referring to the mediator complex. For the mediator, why do they form just 2 foci per cell in the DMSO control?
Yeah, it's a very good observation. So on the quantification plot on the left, you would have seen that there's a bit of a spread. So we see cells where we see up to 7 or 8 foci, and we cells where we only see limited number of foci, like 1 or 2. I think we would have expected more. I think some of that has to do with the limitation of our microscopy setup. And one thing to consider is also that these are haploid cells. So probably we see, just based on that karyotype, we see a lower number of foci. But I think in the big picture, it was within the range of what one expects from these very large foci. So I think our experimental setup lacks the resolution to capture the smaller foci that were reported also in the literature. But it's, whoever asked the question is also perfectly right. We were also interested / surprised by the fact that, in most of those cells, we saw only a handful of those foci. Also, it's fair to point out that this is a microscopy on fixed cells, so it's not live cell microscopy. So it's really always a snapshot in time, what we're seeing here.
Okay, fair enough. Other question we have here is, for the degradation of specific transcription factors or oncogenes in cancer cell lines, which approach do you think is the most suitable?
So I think it all comes down to whether you have a trustworthy ligand binding site in that particular transcription factor. And I think for the vast majority of transcription factors, that might not be trivial, but there's always exceptions. So there's 2 exceptions that I can mention. For instance, STAT family transcription factors, such as STAT3 or STAT5, obviously have an SH2 domain, which is very important for their activity, and which has been shown both for inhibitors, that you can exploit that pocket for inhibitor design, and there has also been very exciting work by Xiaoming Wang's lab showing that we can, or that they can synthesize highly active STAT3 PROTACs that were also in vivo active. And there's also companies trying to push STAT3 degraders into the clinic. So that would be exception 1. There's always transcription factors that have ligandable pockets. And exception 2 is that there's, of course, also a lot of very exciting movements in the covalent ligand discovery, where it has been shown that multiple also unstructured domains that we would traditionally consider as non-ligandable could be liganded with covalent ligands. So that could still be a strategy. It again comes with a little asterisk that a covalent ligand won't be likely as effective to induce protein degradation, because in all likelihood you're losing at least certain aspect of the catalytic turnover. But this is also a very active field of research where we are not part of, but yeah, there's a lot of exciting literature.
Okay, great, thank you. Is it common to observe increased mRNA levels of the target protein to compensate for PROTAC-driven degradation?
Yeah, it's an excellent question. This is something that we would be super excited to explore on a very systematic level. We have observed it here and there. We have also observed it in just the literature. I think, for instance, the degradation of arbrin39 via these aryl sulfonamide glues is one example where you see that mRNA levels go up as a compensation mechanism. I'm not sure how often that is the case on a really, if we would study that systematically. And in those cases where we are aware of such a feedback loop, the increased mRNA synthesis kind of never catches up if the degrader that we are using is catalytically potent enough. So I think in those cases where I'm aware of it, it's kind of a last effort that the cell lays out in order to circumvent protein degradation. And more often than not, the PROTAC wins.
Okay. I think this is a more general question. Is global protein degradation affected by UBE2M? And do the cells experience translational stress when factors of the protein degradation machinery are knocked out?
So I think this is a bit of a two-pronged question, if I understood it right. So I think
correct.
The first question would be if we see some sort of global proteome alterations in these UBE2M hypomorphic mutants. And the answer is we don't know. I think we have not yet done comprehensive expression proteomics comparing wild-type cells to these mutants or mutants to the reconstituted clones. So I don't know, but I would strongly suspect that to be the case. And then the second question is goes along the translational stress. To be honest, I don't know. We have never looked into that.
All right. Okay. How surprising was it to you that multiple labs identified cycling K molecular glues?
Yeah, it was very surprising, to be honest, because typically whenever you're on the receiving end of 3 manuscripts that all come out at the same time, you kind of anticipate that this can't be true. And then when you're on the other end and you realize that 2 other labs have made very congruent and very similar observations, you have to accept that this can happen.
Yeah, right. Just
so, but why that was the case, I think it might be the result of a heightened awareness of these sort of molecular glue degraders or non-obvious ligand-induced degradation events. And I think there will be a lot of exciting data still to be discovered how it can be that chemically diverse substances can induce the degradation of the same protein via the same mechanism.
Okay. Coming back to the mediator, does splicing efficiency change after mediator loss?
It's a very good question, and we haven't studied it.
Okay.
If the person asking the question is interested, I'm happy to provide cell lines and tools. Very
good. Can you say a little more about the DTAG technology in general? So does targeted degradation via DTAG work for every protein?
Yeah, it's a good question. So I think in our hand, where we are mostly interested in gene control factors, it typically tends to work. But we also have, this was a paper or work out of Giulio Superti-Furger's lab here at CeMM, where they are interested in solid carrier proteins. So these are transporters that are multi-pass membrane proteins that can reside either in the cell membrane or in membrane of different organelles, such as mitochondria. And so they have done a very systematic survey where they detect different SLC transporters. And they have clearly seen that there are some compartments where at least cereblon-induced target protein degradation fails to induce productive proteolysis. So, for instance, SLCs that are lining the mitochondria were always kind of refractory to induce degradation. And the reason for that is that likely the ligand binding site was at the interior of the mitochondria where there's simply no cereblon available. So I think with cereblon-based technology, I think it's also worth mentioning at this point that there's been a very recent extension published by Benam Nabed from Nathaniel Gray's lab, where now also the VHL ligase can be hijacked. And I think that is a very beautiful complementary strategy to the cereblon-based DTAG method. And in fact, we have also used that in our Mediator paper because there's also cell lines that appear to be a bit more refractory to cereblon-based induced degradation. So for instance, we struggled initially to induce degradation of our mediator proteins in these HCT 116 colorectal carcinoma cells. So there we knew it couldn't be the protein because the same protein with the same tagging site worked in our leukemic cells. And so it was an easy fix by just switching the ligand from a cereblon-based degrader to a VHL-based degrader, and then it worked very nicely.
Okay, well, thank you. It looks like this is all the time we have for questions today. So let me thank our speaker, Dr. Georg Winter, and our sponsor, Lexogen. And if we didn't have time to get to your question, we will try to have somebody follow up with you via email. Also, as a reminder, again, please look out for the survey after you log out to provide us with your feedback. And if you missed any part of this webinar or you would like to listen to it again, a link to an archived version will be emailed to all attendees. So with that, thank you very much again for attending this Genome webinar.
Thank you.